Welcome back to the Deep Dive. Today we are tackling, I think, one of the biggest challenges in neuroscience. It's this question of how damage at the microscopic cellular level, how that actually becomes the large-scale network problems we see in conditions like autism or schizophrenia.
Exactly. We're talking about a concept called connectopathy.
Connectopathy, right. The idea that these disorders are fundamentally about altered or, atypical brain connectivity.
And the mystery has been there for 3 decades now. We have these huge data initiatives. I mean, you're talking Abide, Enigma, the UK Biobank. These have given us rock-solid proof from human fMRI that this altered connectivity exists.
So we know what's wrong, at least at that macro scale.
We know what's wrong. We can look at a scan and say, that connection looks weak.
But what does weak actually mean?
That is the multi-million-dollar question. That is the explanatory gap. We see the network is, let's say, running slow, but we can't tell why. Is it a broken traffic light at the synapse? A problem with the excitatory and inhibitory signals? Or is the road itself paved poorly, which is more like a developmental miswiring problem?
Until we can bridge that gap, those, beautiful fMRI brain maps are just showing us symptoms, not the cause.
They're just symptoms.
Exactly. So, okay, if human research gives us the what, we need something totally different to get at the how and the why. And that, according to the sources we're digging into today, is where rodent fMRI comes in.
It is our causal testing ground. In a rodent model, we can do targeted perturbations. We can go in and test the exact mechanism we think is broken. That's something that's just, it's impossible to do in humans.
And you can link it all back to the huge amount of genetic and molecular data we already have for the mouse brain.
Yes, you can truly connect the dots from the cell all the way up to the network.
Let's get into the technical side, though, because this didn't just happen overnight. 15 years ago, trying to map these networks in mice, the results were all over the place.
Oh, absolutely. It was a mess. They needed a convergence of things, much better MR hardware, much tighter control over the animal's physiology, like their breathing, their temperature, and really critically, better ways to analyze the data to get rid of motion.
And when all that finally clicked into place,
a second wave of studies confirmed it. We can, in fact, reliably identify these resting state networks, or RSNs, in rodents.
But the real key, it seems, wasn't just the tech, it was people deciding to work together.
That was the game changer. About 2 years ago, 17 different labs from all over the world basically said, okay, let's stop arguing about methods. They built a common standardized pipeline for processing mouse fMRI data.
And it worked?
It worked. They found multiple reproducible RSNs, including a really solid rodent version of the human default mode network, the DMN. And that shared roadmap is what makes all the future work comparable, translatable.
Okay, so speaking of technical hurdles, let's talk about the elephant in the MRI suite. How do you get a mouse to stay perfectly still? Immobilization is everything for fMRI, but the methods, they sound pretty controversial.
It's a huge balancing act. For a long time, most studies just used light anesthesia, something like midazolam isoflurane to make sure the animal didn't move at all.
But the problem there seems pretty profound. Anesthetics are drugs.
They are powerful pharmacological agents. They can mess with neurovascular coupling, which is the very thing fMRI measures, the link between neuron firing and blood flow. Or they can have weird interactions with the specific genes you're trying to study.
So the anesthesia creates this kind of paraphysiological state, as the sources call it. It's not a natural brain state.
It isn't. And that's why the field is pushing so hard now toward what we call the awake standard.
Awake fMRI. But that sounds incredibly difficult.
It is. The goal is to minimize stress. And that means you need these really rigorous, repetitive habituation protocols.
So you're talking weeks of training just to get one clean signal from one mouse.
Weeks. You are literally training the animal day after day to be comfortable and calm inside the scanner. But when you do it right, their stress hormones, their corticosterone levels, are basically the same as if you were just gently holding them.
So that's the gold standard now.
Awake imaging is the gold standard. Anesthesia is still seen as acceptable, but you have to prove it's not interfering with what you're studying.
This sets up the big question perfectly then. We know we can map these networks reliably, but how much does a mouse's brain map actually look like ours?
And that takes us to the field of comparative functional neuroanatomy. looking at brains from an evolutionary perspective.
Mhm.
And the news here is actually pretty encouraging. We see strong homologies.
Meaning comparable architecture.
Exactly. Comparable architecture between rodents and humans in some really key systems. The salience network, the motor network, limbic systems, and of course that default mode network.
But where does it break down? Where do the maps not line up?
The divergence is just as important. The big one is the DMN hub. In you and me, the DMN is anchored by a region called the precuneus. It's this huge hub that's involved in, wow, really high-level stuff. Memory, imagination, thinking about yourself.
And mice just don't have that.
They don't have a clear anatomical equivalent. It seems like its function is sort of transferred to a different area, the retrosplenial cortex.
So the mouse DMN works, but it's wired differently. That's a huge caveat for translation.
It's a critical caveat. And the other big one is certain primate-specific circuits. For example, researchers tried to match the connectivity coming from parts of the human striatum areas involved in executive function, even language. They just couldn't find a match in the mouse brain.
Okay, so we have to be careful. The basic architecture is there, but the higher order stuff is distinctly primate.
Precisely.
Which makes this next step so fascinating because now, with that groundwork laid, we can use this controlled rodent environment to really test the origins of the disconnectivity we see in humans. This is where we start closing that explanatory gap.
This is it. The value proposition is total control. We get around all the problems in human studies, patient motion, genetic differences, different life experiences. Here, we can use genetically identical litter mates, matched for age and sex, in a completely controlled setting.
Let's dive into the first paradigm, genetic modeling. What happens to brain connectivity when you isolate one specific disease-related gene?
The best work here has really been in autism spectrum disorder, or ASD. They've looked at a whole host of risk genes, SHANK3, fMR1, TSC2, and a big one, the 16p11.2 microdeletion.
And they found direct links, right? Things they could actually explain mechanistically.
They did. With the 16p11.2 microdeletion, the mice showed the exact same prefrontal hypoconnectivity, so weaker connections, that you see in humans with that deletion.
Wow.
Yeah, and then in the mouse, they could trace it back to the cause.
Rah.
Immature wiring between the thalamus and the prefrontal cortex. It was a huge win.
And I saw another one with the TSC2 gene, which caused hyperconnectivity, which, again, mirrors what's seen in some autism patients.
Exactly. These were massive validations for the whole approach.
But then a huge study came along, looked at 16 different mouse models of autism, and kind of threw a curveball into the mix.
It was a vital finding, a reality check. They went in looking for a single converging autism network, and they didn't find it.
What did they find instead?
A huge, broad array of abnormalities. And often, they were completely distinct signatures. Sometimes they were even pointing in opposite directions across the different genetic models.
Wait, so you're telling me that we shouldn't be searching for the autism network signature? We should be looking for subtypes of broken networks.
That's what the evidence supports. It's a reconceptualization. The disconnectivity you see is dictated by the specific underlying biological problem. It's not one size fits all.
That changes everything about how you'd approach diagnosis and treatment.
It does. And it leads us right to the second approach, which is not just about genes, but about pathophysiological process modeling. So looking at general mechanisms like chronic stress or problems with how synapses are pruned during development.
And this is where it gets, frankly, mind-bending. Decoding via multimodal fMRI. This is where they can literally flip a molecular switch in a mouse's brain and watch what happens to the fMRI signal on a macro scale.
This is how we get to reverse translation. We use these incredible tools, chemogenetics, which are often called DREAD-Ds, and optogenetics, to link a very specific microcircuit change, like the famous EI imbalance hypothesis, directly to a signature we can see on an fMRI.
Okay, let's stop there. EI imbalance. Break that down for us.
Sure. Think of your brain as an orchestra. The excitatory neurons, the E, are all the instruments playing notes. The inhibitory neurons, the I, they're the conductor, making sure everything is in harmony and on tempo.
So if the conductor loses control?
You just get neurological noise. It's a leading hypothesis for what's going wrong in a lot of disorders.
So they manipulated this conductor in mice and watched the fMRI. What happened?
They chemogenetically boosted the E relative to the I in one specific area, and just as they predicted, the overall fMRI connectivity in that cortex went down. But here's the crucial part. They measured a very simple metric from the signal called the Hurst index.
Okay, plain English, forget the 4F-POLD signal statistics. What's the Hurst index?
Think of it as a measure of the memory or the predictability of the brain's internal chatter. Does what's happening right now strongly predict what's going to happen in the next moment? They found that when they messed with the EI balance, this chatter speed measurement changed in a very predictable way.
So manipulating the EI balance gave them the calibration key.
It gave them the key.
And with that key, they could go back to the human clinical data.
And decode it. That's the reverse translation. They used the Hurst index signature they'd calibrated in mice to find regions of likely EI imbalance in human patients. And it worked. It pointed right to impairments in specific social brain regions, like the medial prefrontal cortex.
That is genuinely incredible. So we can use the mouse model to infer something about the precise microcircuitry from a fuzzy human brain scan.
That's the power of this approach.
Okay, so just to recap this section, the big takeaway is that connectivity changes are heterogeneous, they depend on the specific cause, but we have these amazing tools that let us translate micro-level pathology, like EI imbalance, into something we can actually measure and decode at the macro level.
Exactly. And now that lets us shift from thinking about static wiring to how the brain actually achieves its dynamic flexibility.
I love this final section. We're talking about the neuromodulatory systems, the NMSs, serotonin, dopamine, norepinephrine. These aren't the main data highways, are they? They're more like the global setting suggester.
They're the adaptability layer. They are what allow the brain to switch states, from quiet internal thought to being highly vigilant and focus on the outside world. If those systems are dysregulated, the brain gets stuck. It loses its flexibility, and that can lead directly to psychiatric symptoms.
And the first attempts to study this used what's called pharmacological fMRI, right? You just give someone a drug like ketamine and put them in the scanner.
Right, pHMRI. But it was an incredibly noisy signal.
Why?
Two big reasons. First, off-target effects. The drugs hit more than just the system you're interested in. And second, and this is worse, direct vasoactive effects. The drugs themselves make blood vessels constrict or dilate, especially outside the brain.
So the signal you're measuring isn't the brain activity, it's just the drug's effect on plumbing.
To a large extent, yes. And we have a perfect example of this in the sources.
Right, the serotonin one.
Yes. When researchers used a clean, targeted chemogenetic tool to activate just serotonin cells, they saw specific fMRI activation in memory and reward circuits. But when they used a traditional pharmacological drug to boost serotonin everywhere, they saw widespread fMRI deactivation. It was mostly just peripheral vasoconstriction. The clean tool gave the real neural signal. The drug gave vascular noise.
So again, it's opto and chemogenetics that give us that clean signal, letting us map what these modulatory systems are actually doing endogenously, and what have they revealed?
They've confirmed that these systems profoundly shape connectivity by shifting the entire brain's dynamics between what we call synchronous and asynchronous states.
Okay, give us some analogy for synchronous versus asynchronous.
Think of a stadium crowd. When the brain is in a synchronous state, it's like the whole crowd is chanting the same thing, very loudly. You have high global connectivity, but the crowd isn't really listening for any specific external sounds.
Okay.
When the brain is in an asynchronous state, it's like everyone in the crowd is having their own quiet individual conversations. Global connectivity is lower, but if someone suddenly yells fire, they can respond much more quickly and selectively.
So neuromodulators are basically controlling whether the crowd is chanting together or having lots of separate conversations, whether the brain is globally integrated or locally focused.
That's a great way to put it. Take the locus coeruleus norepinephrine system. It's tied to stress and arousal. When you activate that system, it pushes the brain into a more synchronous integrated state. It literally increases low-frequency connectivity in networks like the DMN.
And they could link that directly to the hardware.
They did. The strength of that change was physically correlated with the density of norepinephrine receptors in those brain regions. It's this beautiful link between a global stress signal, the receptors on the cells, and a dynamic change in the entire network state.
This has been an amazing deep dive. We've gone from these big blurry pictures of dysconnectivity in humans all the way down to flipping single switches in mice to understand the cause. Rodent fMRI is really letting us close that explanatory gap.
And the future is moving toward longitudinal and awake imaging. So tracking the effects of some genetic or environmental factor over the whole lifespan of the animal, and constantly rigorously comparing those animal maps to high-quality human data. That's the only way this works.
And ultimately, the goal isn't just to find a biomarker. It's to understand the biology so well that we can do that reverse translation. that we can look at a clinical fMRI scan from a patient and decode something precise, like an EI imbalance in a specific circuit.
That ability to link the molecule to the mind. That's the whole promise of the field.
So we now know that our fundamental ability to be flexible, to switch between focused attention and quiet rest, is controlled by these dynamic neuromodulatory systems. So here's a final thought for you to chew on. Given how varied and complex psychiatric disorders are, should we maybe be focusing our search for treatments more on correcting these flexible dynamic systems, rather than trying to fix what we think of as static, broken wiring? Something to consider until our next deep dive.